Sun is the source of almost all energy that sustains life on Earth. All organisms need food, but only green plants are capable of trapping solar energy to manufacture food from simple substances. The process by which chlorophyll pigment in the plants absorbs sunlight and utilize it to make food and oxygen from carbon dioxide and water is called photosynthesis. Photosynthesis takes place in the leaves of the plants, and so leaves are regarded as nature's food factories. Sun sends us energy in the form of small bundles called photons. Photons group together in the form of waves which have different lengths or different number of photons. The sun gives off a full range of light of varying wavelengths called the electromagnetic spectrum. The electromagnetic spectrum includes gamma rays, X-rays, UV rays, visible light, infrared light, microwaves and radio waves. Photons at shorter wavelengths like UV rays tend to be very energetic and can be damaging to the cells and tissues. Fortunately for us, they are mostly filtered out by oxygen or ozone layer in the stratosphere. Photons at longer wavelengths like infrared light and heat rays do not carry enough energy to allow photosynthesis to take place. The radiation in between these two extremes, referred to as visible radiation, is mostly used for photosynthesis. After the filtration of short wavelengths, the leftover radiation consists of about 4% UV, 52% IR or weaker light and 44% visible light. All of this light is available to plant for use, but they only use visible light in the process of photosynthesis. Photosynthetically active radiation, often abbreviated PAR, designates the spectral range of solar radiation from 400 to 700 nanometers that photosynthetic organisms are able to use in the process of photosynthesis. PAR is normally quantified as micromole photons per meter square per second, which is a measure of the photosynthetic photon flux density or PPFD. Other living organisms such as green bacteria, purple bacteria and heliobacteria can exploit solar energy in slightly extended spectral regions such as the near infrared. These bacteria live in environments such as bottom of stagnant ponds, sediments and ocean depths. Because of their pigments, they form mats of green, red and purple color. Plants make an amazing variety of pigment molecules, far more than animals. They sense light to control their growth and rapid responses to the environment, besides using light as their source of energy. Plants can carry out photosynthesis because they contain a pigment called chlorophyll. Now, what are pigments? Pigments are molecules that absorb specific wavelengths or energies of light and reflect all others. Photosynthetic pigments are present in the chloroplast of green plants or in the cells of photosynthetic bacteria and capture the light energy necessary for photosynthesis. Pigments are colored. The color we see is the net effect of all the light reflecting back at us. The reflected light is the incident light minus the light absorbed by the pigment. Thus, the green color of the leaves is due to efficient absorption of blue and red light, but not green by the chlorophyll pigment. The transmitted green light gives the leaves their green color. The color of the leaf depends on the type and quantity of pigment it has. During winter, there is not enough light or water for photosynthesis. The trees will rest and live on the food they store during the summers. They begin to shut down their food making factories. The green chlorophyll disappears from the leaves. As the bright green fades away, we begin to see yellow and orange colors. Small amounts of these colors have been in the leaves all along. We just can't see them in the summer because they are covered up by the abundant green chlorophyll. Several kinds of pigments are present in plants and prokaryotic photosynthetic organisms. For example, chlorophyll A, chlorophyll B, chlorophyll C and D, bacteriochlorophyll A, bacteriochlorophyll B, C, D, etc. and carotenoids. Each pigment absorbs different wavelength of solar radiation and covers most of the solar spectrum. For example, chlorophyll absorbs at 430 nm and 630 nm. Types of pigments involved in photosynthesis. Major and accessory pigments. A major or core pigment like chlorophyll A is the one which is present at the reaction center and takes part in the primary photochemistry or oxidation reduction reactions induced by light. The accessory or peripheral pigments like chlorophyll B and carotenoids only collect light and transfer it to the reaction center and does not carry out any photochemical reaction on their own. Chlorophyll A. This is the most abundant pigment in plants. Chlorophyll A absorbs light with wavelengths of 430 nanometers in the blue region and 660 nanometers in the red region. It reflects green light strongly, so it appears green to us. The molecular chemical formula for chlorophyll A is C55 H72 N4 O5 and 1 magnesium. Structurally, chlorophyll has two parts, a tetrapyrrole ring and a phytol tail. A magnesium atom at the center of the planar portion is coordinated to 4 nitrogen atoms. The nitrogen atoms are each part of a pyrrole, which is a cyclic organic compound with a nitrogen atom in a 5-member ring with 4 carbons. For this reason, chlorophylls and related compounds are often referred to as tetrapyrroles. A 5th ring is formed at the lower right corner and a long hydrocarbon tail is attached to the lower left. It contains a hydrophobic, fat-soluble phytol tail that allows it to be embedded in a lipid membrane. The rest of the structure rests outside of the membrane. It is the tetrapyrrole part of the pigment that absorbs the energy from light. The metal at the center of the structure, magnesium, can have variable oxidation states. This means that it can accept and donate electrons readily depending on the situation. It is flexible, which is very important for the function of the molecule. Most of the chlorophylls are classified as chlorines rather than porphyrins by virtue of the reduced ring D. Chlorophyll biosynthesis begins with the formation of 5-aminolevulinic acid, that is ELA, and the biosynthetic pathway involves 17 enzymatic reactions. Chlorophyll B. This molecule has a structure similar to that of chlorophyll A, except for a formyl group at C7 instead of methyl group. It absorbs light of 453 nanometer and 642 nanometer maximally. It is not as abundant as chlorophyll A and probably evolved later. It helps to increase the range of light a plant can use for energy. It occurs only in green algae and green plants. Besides these two chlorophylls, chlorophyll C, D and F are also present in some photosynthetic organisms. Chlorophyll C is most unusual of all chlorophylls in not having a phytol tail and also does not have D ring reduced. It is found exclusively in a group of marine algae such as diatoms and dinoflagellates. Chlorophyll D is a structurally and energetically intermediate between chlorophyll A and bacteriochlorophyll A, suggesting that it may have been an intermediate between the transition from anoxygenic to oxygenic photosynthesis. Carotenoids. This is a class of accessory pigments that occur in all photosynthetic organisms. They are completely hydrophobic, that is fat soluble, and exist in lipid membranes. Carotenoids absorb light maximally between 460 nanometers and 550 nanometers and appear red, orange or yellow to us. The spectacular colors of fall foliage are due partly to the yellow-orange light reflected from carotenoids. Some of these accessory pigments, in particular the carotenoids, also serve to absorb and dissipate excess light energy or also work as antioxidants. Carotenoids belong to the class of tetraterpenoids, that is, they contain 40 carbon atoms being built from 4 terpene units. Structurally, carotenoids form an extended Pauline hydrocarbon chain which is sometimes terminated by rings and may or may not have additional oxygen atoms attached. Xanthophylls, beta carotene, lutein are some examples of some other accessory pigments. Phycobilisomes are blue-green and red pigments present in cyanobacteria and red algae. They are light harvesting antenna of photosystem II in cyanobacteria and red algae and absorb in the spectral region from 550 nanometers to 650 nanometers. Each phycobilisome complex consists of 2 or 3 types of pigment proteins known as biliproteins along with a number of additional proteins called linkers. Bilins are linear open chain tetrapyrrole pigments. The biliproteins are usually of 3 types: phycoerythrin, phycocyanin and allophycocyanin, which differ in protein identity, chromophore type and attachment. Their relative location in the architecture of the phycobilisome complex is also different, where phycoerythrin is located at the ends of the rods, phycocyanin is located between the ends of the rods and the core, while allophycocyanin forms the core of the phycobilisome. Why there are so many types of pigments when actual photochemistry is done only by one major pigment, that is chlorophyll A in higher plants? The different chlorophyll and non-chlorophyll pigments associated with the photosystems have different absorption spectra because of intrinsic structural differences. The antenna pigments are predominantly chlorophyll B, xanthophylls and carotenoids. Chlorophyll A is known as the core pigment. Their absorption spectra are non-overlapping in order to broaden the range of light that can be absorbed in photosynthesis. The photosynthetic action spectra match with the absorption spectrum of pigment molecules. Now, what do I mean by absorbance? Electrons in molecules can exist at specific energy levels. Normally, they exist at the lowest possible energy level they can. However, if they get enough energy, they can absorb that energy and occupy higher energy level. This is what pigments do. The light they absorb contains just the right amount of energy necessary to push them into the next higher level. Any light that does not have enough or has too much energy cannot be absorbed and is reflected. The electron in the higher energy level, however, does not want to stay there, that is, it becomes unstable. It wants to return to its normal lower energy level. In order to do this, it must get rid of or release the energy that has put it into the higher energy state to begin with. This can happen in several different ways. First, the extra energy can be converted into molecular motion and lost as heat. Some of the extra energy can be lost as heat energy, while the rest is lost as light. This re-emission of light energy is called fluorescence. The energy, but not the electron itself, can be passed on to another molecule. This is called resonance. Lastly, the energy and the electron can be transferred to another molecule. The third and the fourth options are important for photosynthesis to take place. The pigments, along with some proteins, constitute the antenna complex. An antenna complex or the light harvesting complex LHC is an array of pigment protein molecules that absorbs light. Each antenna complex has between 250 to 400 pigment molecules, and the energy they absorb is shuttled by resonance energy transfer to a specialized chlorophyll protein complex known as the reaction center of each photosystem. Transfers are very fast, happens in picoseconds, and the molecules have to be within a certain distance of each other, approximately less than 10 nanometers. In plants, light harvesting complex associated with photosystem 1 is called LSC1, while if it is associated with photosystem 2, it is named LSC2. The purpose of these photosystems is to collect energy over a broad range of wavelengths and concentrate it to one molecule called as the reaction center, which uses the energy to pass one of its electron onto a series of molecules that form an electron transport chain. In this simplified figure, energy is absorbed from a photon of light and transferred stepwise to the reaction center P700. In the reaction center, the energy is imparted to an electron, which is passed on to a chain of carriers. The reaction center initiates a complex series of chemical reactions that capture energy in the form of chemical bonds. The reaction center is thus able to get rid of the energy by passing the energy and its electron onto a series of molecules. The reaction center is the only molecule which can relieve the photosystem of the excess energy. This means that all other pigments will pass the light energy through resonance until it reaches the reaction center. There are two kinds of photosystems in most photosynthetic eukaryotes. When working together, they absorb enough energy from the sun to split a molecule of water. Photosystems are functional and structural units of protein complexes involved in photosynthesis that together carry out the primary photochemistry of photosynthesis, that is, the absorption of light and the transfer of energy and electrons. They are found in the thylakoid membranes of plants, algae and cyanobacteria. In plants and algae, these are located in the chloroplast, while they are located in cytoplasmic membrane in case of photosynthetic bacteria. There are two kinds of photosystems, photosystem 2 and 1. Photosystem 1 probably was the first to develop and can work either with or without association with photosystem 2 to create energy for a plant. Photosystem 1 consists largely of chlorophyll A molecules and contain no or few chlorophyll B. Its reaction center, a molecule called P700, absorbs light of 700 nanometers maximally. Photosystem 2 is the second photosystem to develop in higher autotrophs. It works together with the photosystem 1 to absorb enough energy to split water molecule into oxygen, proton and an electron. Remember, this is the first half of the photosynthetic light reaction where water is split into oxygen, electron and protons. Photosystem 2 contains both chlorophyll A and chlorophyll B. Reaction centers. Reaction centers of plants and bacteria are the world's most efficient solar battery where light energy is converted into chemical energy. These are extremely efficient, showing quantum efficiency of 90 to 95% and rapid which work within a few picoseconds and work efficiently even at low temperatures. A photosynthetic reaction center is a complex of several proteins, pigments and other cofactors assembled together to execute the primary energy conversion reactions of photosynthesis. Two families of reaction centers in photosystems exist. Type 1 reaction centers such as photosystem 1 in chloroplast and in green sulfur bacteria, and type 2 reaction centers such as photosystem 2 in chloroplast and in non-sulfur purple bacteria. Type 1 photosystems use ferredoxin like iron sulfur cluster proteins as terminal electron acceptors, while type 2 photosystems ultimately shuttle electrons to a quinone terminal electron acceptor. One has to note that both reaction center types are present in chloroplast and cyanobacteria, working together to form a unique photosynthetic chain able to extract electrons from water, creating oxygen as a byproduct. A reaction center is laid out in such a way that it captures the energy of a photon using pigment molecules and turns it into a usable form. Once the light energy has been absorbed directly by the pigment molecules or passed to them by resonance transfer from a surrounding light harvesting complex, they release 2 electrons into an electron transport chain. After having cleared the concepts of pigments, photosystems and reaction center, we will discuss the electron transport chain in the next module.